So unlike other metabolic pathways like glycolysis or the citric acid cycle, where we went through them in excruciating detail, understanding each step, the substrates, the products, the enzymes, rate determining steps, looking at enzyme active sites, we're not going to do that for the bulk of chemistry that we see here with fatty acid metabolism. But we are going to sort of have this 30,000-foot view of what happens with beta oxidation. Remember, beta oxidation is the breakdown of fatty acids. It occurs in the mitochondria, and we're going to talk about the 4 steps, just very generically, as oxidation, hydration, oxidation, and cleavage. And so I don't even have structures of molecules here because I just want us to be able to think about this in a very high level, but we are going to consider some sort of functional group interconversions, and I'm going to show you how it's got homology to things that we've already studied. So just looking at this picture first, and then we'll put it together with some words, here is an 18-carbon fatty acid that has been activated so that we've got CoA on it. It's been transported into the mitochondria using the carnitine transport system, and then we've got this now as fatty acyl-CoA that's ready to undergo beta oxidation. So importantly, I want us to look and see that there's a difference between the different ends of this molecule, right? When we talked about glycogen metabolism, we knew how that happened. We had to chew in from non-reducing ends on a glycogen polymer. These are much smaller molecules, and clearly they have 2 ends, but the 2 ends are very different, right? And if we look at the end that we have here of a fatty acid, really there's no functional groups that are there. So there's not much chemistry we're going to do from this end. I'm going to be calling this end of the molecule the business end, and that's because that's where we have functional groups. So this is going to be the business end of our molecule. So highlighting, this is where we've got our functional groups, okay? Reminding ourselves of what we have here. So here's CoA, okay? There's a carbonyl, so we've got our thioester that we have here. And I'm going to be labeling these carbons. This is going to become important here. This is the alpha carbon and the beta carbon. So when you're looking at a carbonyl containing functional group, the carbon that's immediately adjacent is called the alpha carbon. The next one is called the beta carbon. Okay, so what we're going to see here is functional group changes that we have here. We start out with an alkane. Okay, and I'm going to color code these because we're going to use that when we start talking about these in just a second. Okay, so in red there, we've got those 2 carbon atoms, fully saturated CH2, CH2. This is an alkane. Okay, what we're going to see here is we are going to see oxidation chemistry. Okay, FAD becomes reduced as FADH2, so our substrate must become oxidized, and we can see that here. We've introduced a double bond, so we have made an alkene. Okay, I'm going to color code this here. We're going to use orange now. We're going to go through the colors of the rainbow here. So now we went from alkane to alkene. Okay, now we're going to have hydration. So we're going to add water. We're going to hydrate an alkene. Hydration of an alkene, it does happen regiospecifically, so we're going to see the production of an alcohol. We're not going to get into any details about how that regiospecificity happens or even the stereochemistry that we have here, but we're going to generate an alcohol, okay? And so here you see those functional groups. Then we're going to do another oxidation chemistry. Remember, NAD plus going to NADH. That's reduction of that B vitamin, so we must have oxidation of our substrate. And indeed, we go from an alcohol to a ketone. Okay, so there is again our ketone. One of the things that's really unique here, we're going to highlight this in a minute. Again, this being our alpha carbon and our beta carbon, we have a beta keto thioester. A beta keto thioester has lability, has a bond that's easily broken between the alpha and the beta carbons. So the next step that we see is cleavage. And when we break that bond, okay, what we're going to generate is an acetyl-CoA molecule, a C2 onto CoA. So this is our acetyl-CoA. Okay, and this chemistry happens such that we generate another fatty acyl-CoA that's simply shortened by 2 carbon units that come in from the business end of the molecule. So what we do then is we are just going to repeat this chemistry. Oxidation, hydration, oxidation, cleavage. We're going to keep doing that, snipping off 2 carbon units at a time as acetyl-CoA until all of our fatty acyl-CoA has become acetyl-CoA. So let's take a second and review then what those functional group interconversions are, right? So we started out with CH2CH2, right? We had that as an alkane. Undergoes oxidation to generate an alkene. That was our guy in orange here. Alkene. We do hydration. Okay, we're going to hydrate that alkene. We get an alcohol. Okay, that was in orange here. I'm sorry, in yellow to get to our alcohol. And then finally we do another oxidation step and we get to our ketone. Okay. Zooming out for a second just so we could see that sort of mapped on what we have here. Okay, so one of the themes that we're going to see is when we have alkane to alkene interconversions, that often occurs with FAD, sort of as, I should put FAD, FADH2, depending on the direction that we're having. And then when we have oxidation chemistry between alcohols and ketones, we tend to see NAD plus and NADH with that. Okay, so that's kind of one of those themes that we're going to see. And I want to remind us that we have a homologue of this. We saw alkane, alkene, alcohol, ketone. When we talked about succinate being converted to fumarate, being converted to malate, being converted to oxaloacetate. Again, oxidation, hydration, oxidation, right? So a beautiful homologue that we have here is in the citric acid cycle. That second half of the citric acid cycle was essential for regenerating oxaloacetate to continue that pathway. Last piece I want to just kind of remind us of here is if we look at the structure of a beta keto something, and in this case we have a beta keto thioester. Okay, so we've got a keto group here. Okay, this is the rest of the molecule. You've got a CH2 group. And in this case, we've got a thioester. So when we have a beta keto group that's here, so any type of a beta keto carbonyl containing functional group. So in this case, it's a thioester. Okay. We have lability between the alpha and the beta carbons. So this bond is labile, right? And we've got great videos that sort of show the mechanism of this. This is a reverse Claisen condensation to break that carbon-carbon bond. So really neat ester chemistry that sort of happens when we have reverse Claisen condensation, when we do this cleavage, and then when we actually do fatty acid synthesis, we are going to see a Claisen condensation reaction. Okay, so that is on a very 30,000 foot view, sort of looking at the oxidation, hydration, oxidation, cleavage chemistry that happens with beta oxidation of fatty acids. Again, under a need state, it's going to be happening in the mitochondria, and it's going to generate acetyl-CoA that can either be used locally by taking it onto the citric acid cycle, or if you are the liver, you can convert that acetyl-CoA into ketone bodies, which then can be exported to other cells for use as a hydrophilic circulating fuel.